So, welcome all. I am Shantala Haridas, the Associate Director at IndiaBioscience. And in case your paths haven't crossed with us before, IndiaBioscience is a not for profit organization that works as a facilitator and apologies for that. So, as I was saying, that in case our paths haven't crossed before, India's Bioscience is a not for profit organization that works as a facilitator and a catalyst to strengthen the life science ecosystem in India. And since we have a really jam-packed session today, I'm not going to go into any details of what we do, but do visit our website. The link will be in the chat box, and do participate and engage with us. And one of the things we are very passionate about is highlighting science and increasing the visibility of science in society. And that was a really natural point for us to come together with the Ignite Life Science Foundation and Cactus Communications to co-host this series, which is In Conversations with Ignite. And you'll hear a little bit about this in a few minutes. And so today on National Science Day, we're really thrilled to kickstart this event with a fun and informal, a stripped-down conversation with Ajit Varki. And we're really thrilled to have him as our first guest in this series. He is someone who has worn and continues to wear many hats. He's a physician scientist. He is the co-director, and I'm just going to look to, because the list is quite long. So he's the co-director of something called CARTA, which is the Center for Advanced Research and Training in Anthropology. He is also the co-director of the Glycobiology Research Training Center. And so for those of you who don't know about glycobiology, this is a really fun opportunity to get an insight into that. He is a professor of medicine and cellular medicine at UCSD. He's also the author of a book, Denial. So as you can see, there are a lot of topics to cover in today's conversation. So buckle up for a really fun conversation. And the one thing that I do want to kind of mention to all of you at the start is that please do join in this conversation. Do join in. Do kind of send your questions via the chat box, via the Q&A box, and we'll pick up your questions. If you already submitted your questions at the start of this, at the registration stage, we have collected your questions. We have gone through all of the various topics you have suggested. And Swami, who is the interlocutor for today's session, has woven them into the conversation. And Swami is the CEO of Ignite Life Science Foundation. And so with that, before we hear from Ajit, just a brief introduction into the other co-hosts for today's session. So I'll hand it over to Smita Jain, who is the Associate Director for Academic and Government Relations at Cactus Communications, and also my former colleague. So with that, over to you, Smita.
Thank you so much, Shantala. Welcome, Ajit. Welcome, Swami, and welcome all of you who are here today with us on the evening of National Science Day. It's a great pleasure to be part of this particular series. And I guess we all know, or we all say, that some great ideas do get born over coffee or tea, but interestingly, this idea got born over our conversations online. So that's the power of online these days. Well, I won't take very long. Very quickly, I would introduce Cactus Communications, which is a technology company that's been accelerating scientific advancements. Now, Cactus has been around for about 2 decades, solving problems of researchers, universities, publishers, academic societies, and life science organizations through its vast array of products and solutions, services. Again, that gets offered under its different brands, namely Editage, Cactus Life Science, Impact Science, Researcher.life, and there is a whole list of brands that exist under the Cactus umbrella. Now, Cactus is present across the globe with its headquarters located in Mumbai and a global workforce of over 3,000 people. It's pretty big size organization. Cactus has been also considered a pioneer in its workplace best practices and has consistently been ranked a great place to work over the last several years. And that's, again, something which is very, very inspiring. So that's in brief about Cactus. As I said, I won't take long, but I really wish this series where we are going to have some very engaging, interesting, in-depth conversations with some stalwarts from the scientific community. And we all look forward to these conversations. So over to you, Swami.
Thank you very much, Smita, and join you in welcoming Ajit. Ajit, you need to unmute yourself eventually. Just for everybody's information, Ajit is awake at 6 a.m. his time, so additionally grateful for that, because he kind of adjusted to our request, because 8:30 onwards becomes a little late for us, and we said do it at 7:30, and he was quite accommodating. So thank you very, very much specifically for that. I request everyone who's logged on to stay on till the very end, because we have two very important announcements to make at the end. So please do stay on. And the format for this is pretty informal. I don't have any presentations, neither does Ajit. We are going to be conversing, and he will do most of the talking, I promise you. I will do a little bit. And I hope that over 40 to 45 minutes, we cover a lot of the topics that he has worked on. We will have some time at the end for Q&A. Some of the Q&A questions have been received before, and we'll pick some to ask. And you can also feel free to use the Q&A box to ask additional questions as it strikes you. So Ajit, thank you very much once again for joining us. Now, Ajit is one of those people who is a classical high performer, because it's not that he discovered his new when he was 40 years old. He was a high performer right from the time he was a student at Bishop Cotton. He was the class prefect several years in a row. And then he went to CMC Vellore as a medical student, where he won. I think he swept almost all the medals available through academic competition, including some medals given by the University of Madras, which I had never heard of, because I finished my medicine about a decade after Ajit, but I'd never heard these medals even existed. So that's great start for Ajit and his career. And obviously, people must have been very impressed with what he did. But the question to you, Ajit, is, did you, at that point of time, as a student, medical student, and even maybe as a school student, recognize that you wanted to get into science, wanted to become a researcher?
No, by the way, thank you so much for this invitation, a great honor. So I was always interested in science, but I always wanted to become a doctor. That was my primary goal. And the interest in science came along with my interest in biology and medicine, really. So that's where it started from. And when I went to CMC, again, I was focused on the clinical aspect first, but my research interest then blossomed over there.
So you also did some research while you were at CMC Vellore?
Right. When I was at CMC Vellore, I was very fortunate that that was the time in India when there was very little research going on in most medical schools. But Professor Bachawat, Professor Radhakrishnan, a bunch of people have been recruited to CMC, doing basic research, competing actually with the West. And so I was a medical student, listened to them talk and lecture, and looked into what their science was. But I actually didn't do a whole lot of research myself. I just got inspired by watching them. Mm-hmm. I still wanted to do medicine first. So that came first, and the research came later, but it comes as a package.
And then you moved to the United States. Was that part of the wholesale movement of many medical doctors to the US at that time, or were you having a specific objective in mind?
I had a very specific objective in mind. In other words, I realized after looking at all the literature that a great number of physicians are doing really fantastic science. Mm-hmm. So how does that work? How does the physician who's busy in the clinic actually do research? I realized the United States is one place in the world where there were what are called physician scientists. People who were trained as physicians. Some continued their clinical work, some didn't. But they applied their medical knowledge to very basic research, but also got trained very well in basic research. Yeah. So I decided the only way to do that is to go to the US and train in basic research. So my intention of going to the US is not to get a position in practice or in academia. I wanted to do research. But I finished my clinical training also there.
Okay. So, I mean, it's good that you were convinced at a very deep level that you want to do research, because there are many of us who look at a research career with some trepidation, because clearly the money is not very good, especially if you're in India, right? So it requires a lot of guts also to say that I'm going to go into research, I'm going to excel to a level where money does not become such an issue. So I guess you had the confidence in your abilities that you would go to the US, you would get into research, you would shine, and you would do well for yourself.
Well, I wouldn't say I had that confidence. I had the hope to do it, but it was a complete unknown to me going in there, So initially, of course, I was this foreign doctor without much research background. I had to convince people that they should take me on. So
you actually went to a biochemistry lab somewhere on the West Coast.
Yeah, no. First I went to did my medicine, hematology, and oncology at Washington University in St. Louis.
Okay.
Actually, Washington University is in St. Louis, and there I happened to be in the same division, and my division chief was a famous man, Stuart Cornfeld, who's still active today, who really discovered a lot of what we now call the field of glycobiology. And I realized that I wanted to do bone marrow transplantation. That's what I wanted to do. And I started looking at these blood cells and said, what's this stuff? Every blood cell is totally covered with this huge package of sugars, like a forest. How come nobody studies this? There's ABO blood groups, there wasn't much else. So I decided instead of focusing on bone marrow transplantation, I'd first learn about these sugars on the surface of cells. I got into that and never turned back after that. So it also tells you that I started out wanting to be a clinician, then I wanted to be a bone marrow transplant, then I became a physician scientist, now I'm a basic scientist.
Yeah.
The whole spectrum is open to those who are willing to do it.
Amazing. Because the reason I'm said amazing is actually because of what I'm going to say now. One of the first papers, incidentally, was not on carbohydrates, it was on lipids in amniotic fluid, right? So it's interesting. You did work on lipids, but you ended up with Cornfeld working on carbohydrates. And you had a paper in 1980. You graduated with your MBBS in 1974. Am I right?
74, yeah.
And you had finished your internship by 74.
Yeah.
And 6 years later, you were in the US doing research with Stuart Cornfeld, and you had a paper in JBC, genetic regulation of acetylation of erythrocyte sialic acid. Right? So that's quite impressively fast. I mean, normally, if many MBBS doctors from India go to the US, they become pure clinicians, but those choose to do research like you did. Many of them end up getting a PhD, but you did not go that route. You actually went straight into a research lab, Stuart Cornfeld's lab, where he was a glycobiologist, and you just zoomed from there. Am I right?
Yeah, that's how it happened. It so happened at that time in the US, it was not necessary to get a PhD or get anything further. You could go straight to research. And what mattered is what you published and what you found, not what position you had or what titles and degrees you had.
Okay. And you had 4 papers by 1980 in JBC. And would you say that if you had not worked in Cornfeld's lab, you may have ended up not being a glycobiologist? I mean, did glycobiology choose you, or did you choose glycobiology?
Actually, the thing called glycobiology only kind of emerged at the time. So I kind of came into it at the time that it was expanding, basically. And I decided, seeing what's going on there, that I was going to focus on that, but maybe a few other people working in that area. So that's a good area to get into.
Yeah. And I think you continued to publish your work. You worked in Cornfeld's lab for about how many years? 2 years?
3 years.
And then you became an independent investigator, I guess, at the same university.
Right. At that point, I actually tried to come back to India and came back and visited various places. But there was really no opportunity for someone like me who wanted to do research mostly and a little bit of clinical. There's no opportunity. So I decided I had to stay in the US to do that. So then I applied to various institutions in the US and became my first position was San Diego, where I'm still UCSD as an assistant professor in medicine, but doing research.
Okay, I didn't realize that your stay at WashU was that compressed. I mean, I thought it was...
Actually, it was for the total time was 4 years, yeah.
4 years there, okay. Okay, so you are by now doing research, but you are also a clinician, am I right? You have license to practice medicine in the US. I guess you had to take the VQE or something like that. I don't know what they had.
All those various exams,
okay. Okay, so you took all the exams. You got licensed as a board certified physician, as a hematologist, oncologist, right? And you were doing that clinical work, and you also had a research lab. You were running a research lab, and I assume you were applying for grants, NIH grants or something like that, and you were successful, and you were able to fund your own research. What was it like in those days? I mean, you were doing clinical work, and you were doing lab work which was related to the clinical work. You talked about bone marrow transplantation,
but not directly.
But not directly, yeah. So how did you stitch this together into something that was fulfilling for yourself, and at the same time Because many physicians get into research and then they decide to drop their clinical, right? I guess you maintained your clinical practice until
the late 1990s, when I became editor of Journal of Clinical Investigation.
Okay.
It got me so busy that I got out of the clinic and I never really went back. Okay. So I keep in touch. I read and keep in touch with the findings.
So that's a phenotype that's perhaps can be considered as quite common in the US.
It was common. It's important to say it's not so common anymore.
Okay. And that's because of?
So here's an interesting thing. I said, I'm going to go to the US because here's where physicians are doing real research, basic research, right? I thought the US government had this great plan to train all these physicians to go do research. Turned out it wasn't the case. What happened is, from the Korean War to the Vietnam War, there was 25 years when you could get called up for service. In the US, they had the service draft in those days, and physicians are in great demand to send to the wars. The only way to escape was to go to the NIH and do research. So the brightest and best medical students of two generations ran away to the NIH to do US public health service and did research. And there's a string of Nobel laureates and others came out of that, just MDs, who might otherwise have gone to practice. And since then, that's all changed, and it's not easy to become a physician scientist in the US anymore. I was really lucky. I went in a golden time and that was happening, but it happened by accident. It was not a plan of anything by the US government.
And it's difficult because of the time commitments that each part of the thing asks of you, or is it because funding is not generous enough to support such people?
Funding is good for people like that. Once you make it through the initial rounds of proving yourself, then you can do well even as a physician scientist. But yeah, it wasn't the only. But doing the two things together is difficult. But mainly in those days, the physician science would do like 20% clinical. I'd go to clinic 1 day a week, attending rounds 2 months a year, and some other activities. So really 20% was clinical, and 80% was research. That phenotype is very hard to find now in the US anymore, because people are pushed in one direction or the other. Either you finish you focus on clinics, or you go into research. And then if you go into basic research, which you can do, but you completely divorce yourself from medicine, you've lost some component of what makes you special.
Yeah. So the phenotype of the medical scientist, right, the physician scientist, there are some special many of them have been very successful, especially in the US. I mean, MD PhDs or MDs who have gone into research. As you said, the NIH cohort, post-war, intra-war.
They were affectionately known as the Yellow Berets.
Yellow Berets, okay. Is that like a negative connotation?
I just want to say, like saying, yeah, they ran away from the war and went to do research.
Yeah. So I mean, everybody wants to have medical physician scientists. In India now, at the Indian Institute of Science, 425 crores has been donated to create a postgraduate hospital. You must have heard of that, right? The Bakchis have done that. Paksati has done that. And the idea is that they will get physicians and scientists to get close to each other and talk and collaborate and come up with these new ideas. Now, I have a question. To what extent does your clinical practice, I mean, you are seeing patients, to what extent does it really influence your research ideas, and vice versa? To what extent does your research, I mean, in a realistic manner? I mean, theoretically, and it's fancy, nice to say, yeah, you're doing both and you interact and you come up with new, but in a realistic manner, does it really work that way?
Yeah, because once you've been through medical school, medical training, you have a part of the brain that the PhD doesn't have. You know a lot of things that medicine. The medicine is not really a science. Medicine is an art that has a shotgun marriage to science. So a lot of medicine is not logical necessarily. It's traditional, and so I think that the opportunity you have is to cross the bridges between the two. Every once in a while, I see something. I hear another scientist say, "No, that's not how it is. Here's how it really happens in the clinic." So I bring the clinic to the basic scientist. But you said something earlier which is very important to follow up on. You pointed out IISc and CMC and all these other places that do research, but they're all separate. So I'm a visiting professor at NCDS Bangalore and at IIT Madras, and I also visit CMC. Three good institutions. One is primarily technology, the other is medicine, and the other is biological research, and they're separate. In the US, they're all together.
Yeah, co-located on the same campus, yeah.
Yeah, even in the same institution, in the same department, even. And that came because of physician scientist ethos in the US, which is now changing. So,
so I mean, I don't know whether there's any empirical evidence to say that physician scientists have changed the scene for biomedical science, but I think there must be some kind of evidence, which is why people are continuing to pursue that. In India also, it's now becoming fashionable to talk about doing that, although India has not done that. Maybe if this happens, it'll happen, right? So now I want to take a little bit of a orthogonal path and talk about your core interest in glycobiology, right? Now, the audience is a general audience. There are probably not that many glycobiologists in this audience. So if you can give like a 3 to 5 minute overview of the field of glycobiology, what is it? I mean, I know that you have said it is the dark, I mean, you have said in a review article with Confell that you called it the dark matter of the universe. It's kind of, it's out there.
The biological universe,
but in the the biological universe. So it's out there and not many people have studied it. And there may be lots of functions, lots of purposes for this material, but it's still being discovered, and it's hard to study as well. So maybe if you can give us a quick overview,
right.
That would be helpful.
So actually, the analogy of the dark matter is not entirely correct. When it comes to the dark matter of the universe, nobody knows what it is. We still don't know nothing about it. What happened in glycobiology is different. In the first 7 decades of the 20th century, there were Nobel laureates every 10 years for something in glyco, studying sugar. There's DNA, RNA, proteins, lipids, metabolites, and sugar. One biology. Everybody had to learn everything. And what happens about the time I was starting out in the 80s, the big molecular biology revolution hit. Sequencing, PCR, expression, genetics, all that hit. It's so much easier. It's because it's all linear, and there's almost like a intelligent designer behind it. All there obviously wasn't. But on the other hand, glycobiology is extremely complex, very huge. The complexity of glycans is orders of magnitudes over. So natural tendency is everybody to go into the top of the wave, And so that's what happened. So I took a conscious decision at that time, and I said, no, I'm lucky enough to have had this training in this area, which now seems to be disappearing almost. I'm going to stick with it. And so that subset of people like me stuck with it. Then actually Raymond Drake at Oxford came up with the term glycobiology. In some ways, it's sort of, we don't have a thing called proteobiology and nucleic acid biology like this. We have nucleic acid science and chemistry. So in a sense, glycobiology is sort of a way to adjust for the fact that a lot of people in biology don't learn much about glycans anymore. And so it's sort of it's come together now, and more people have started to learn it, and now it's become very prominent. And so basically the bottom line is every cell in nature, without exception, every cell in nature is covered with a coating of sugars. Sort of like it's not the surface of the earth, the surface of the cell is not like Mars, it's like the earth. Nobody studies that stuff. Now which has a huge impact on everything from infectious disease to you name it, it's involved. So, but yet most people don't study it. So that's the dark matter analogy. Now it's changing. People are starting to come into the programs and trying to get people into this field.
Yeah.
But in a sense, it's part of biology. It's molecular biology. It's not, it's not a separate kind of biology.
Right.
So technology is different.
So one of the key aspects is that unlike gene protein biology, where you have a coding for the protein and which is encoded in the genetic information, here you have enzymes which catalytically add sugar molecules to proteins, and the genes that code for these enzymes are the ones that are culprits in many human diseases. Right. So either you have an excessive glycosylation or a lack of glycosylation and patterns of glycosylation.
Right.
And because this is an enzymatic process, it is probably not, the fidelity may not be as high as the fidelity of a gene to protein transcription, because the gene exactly gets made in the protein. I mean, with some errors maybe, but in the case of an enzymatic process, the kinetics of enzymes can vary with lots of conditions. So
even the timing
patterns of glycosylation can change with not just with the time, but with also with the environmental conditions. So that kind of goes to addresses one of the points you make, which is that it's a lot more complicated, right? But is it still very important? I mean, in the sense that if there is so much variation in glycosylation, etc., is it still as important as, let's say, a mutation of an amino acid in a protein?
Yeah, so the way to think about it is this, that it's there in huge amounts in every, so evolution has done the experiment, 3.5 billion years, not a single cell that's not totally covered with sugars, right? So it's there. So what is happening is that the sugars out there, but they're also the contact point for huge numbers of external agents like coronavirus or any other virus or many other diseases. So many things are forcing the surface to change rapidly. You're going to change it, you're going to die, basically. The species is going to die. So very rapidly evolving. So you get a lot more variation between species and so on. And within species also see variations. And that's what makes it very complicated. So another way to think about it is if you think of DNA, RNA, and proteins as being, not proteins so much, DNA, RNA to some extent as being sort of digital. It's not really digital. Yeah. Evolution produces amazing phenomenon. Glycobiology is analog. Everything varies. Yeah. There's no way to nail it down in one place. But if you don't look at it, you miss a huge part of the story.
Yeah, correct. So in fact, that there's another question I had related to the glycobiology is the carbohydrate structure or carbohydrate types different for the mucous membranes, which are exposed to the external world, versus the cell surfaces which are completely internal, let's say the liver-to-liver interaction, liver cell-to-liver cell interaction versus an epithelial cell in the airway or in the gut. Is the mucinome something totally different from what the internal environment sees?
Yeah, there's some general principles, but by and large, there's a big change. And there's also glycolysis inside the cell, which was discovered later.
Yeah.
So on the surface, there's a lot more variation. You can imagine there's an ongoing arms race, right? Various external agents are trying to get you. You're trying to survive, and you've got to keep changing to survive. I'm using terms that sound like somebody's doing it, but in fact, it's evolution taking its course, basically.
Yeah.
So huge amount of diversity.
So if you look at the repertoire of genes that control this kind of carbohydrate modification of proteins, what are we talking about? 10, 20, 50 genes? How are we talking about?
No, there's probably 5 to 10% of the genome involved in this.
Oh, okay.
If you look at the total picture of producing these things, and what's interesting, you go to microbes, even bigger.
Oh.
Many microbes, like 30-40% of the genome consists of things that have something to do with glycosylation, depending on the microbe.
And why do you think that is the case for microbes versus humans?
Because microbes have to revolve very rapidly and escape. Remember, microbes are not only trying to deal with hosts or attack hosts, but there's also phages trying to attack them.
Mm-hmm.
And they bind to sugars.
Okay.
So that's also another, this thing called red queen effect, which is in evolution. People may not have heard this. Nowadays, people don't read Alice in Wonderland anymore. But there's a story where Alice meets the queen. The queen says, "Run." So they run. They're still in the same tree. So Alice says, "Why are we running? You're in the same tree." The red queen says, "I don't know where you're from, but around here, you want to stay in the same place, you better keep running." So there's these cycles in evolution that are going very rapidly. Some things are conserved because they need to be conserved, otherwise you wouldn't have an organism. Okay. And so there's ongoing conflict. So if you change your glycosylation, you're going to change your internal activities too. You can gain advantages and you lose advantages, but sometimes you don't have a choice. So that's sort of how I got into also anthropogeny, because I was looking at differences between species.
Yeah, and talking about differences between species, you have studied the CMAH enzyme mutation. I mean, you have proposed that mutation. Allele repeats in that enzyme coding regions might have incapacitated that enzyme in humans, leading to the accumulation of the precursor. Whereas in other primates, non-human primates, you have the product of that enzyme, and therefore you have a different biology, especially with respect to susceptibility to various infections. Can you just tell us a little bit more about that?
Yeah, so basically what happens is, this gives you an example of why physician scientists may be important. When I was seeing patients in 1984, I saw a patient who was getting horse serum for aplastic anemia, still a treatment was used. The patient had a reaction, serum sickness reaction. So of course, what do you expect? Putting animal proteins into human. Then I looked it up, and people had just found that the reactions against sialic acid, the sugar that I was studying. So how can that be? It's on every cell type. Long and short of it turned out that humans are missing one kind of sialic acid. A mutation that occurred, it affects all humans. And then actually, now we know in other lineages, the birds don't have it either, CMAH. So completely independently that came. So actually the dinosaurs had a mutation in this gene a long time ago. So then it turned out that chimpanzees, our closest evolutionary relatives, that are very similar to us, had the gene. So I realized by accident, it took me 10 years to figure out what was going on. But by mid-1990s, I figured out that this is the first known difference between humans and chimpanzees, generally. Now there are many hundreds known, but it turns out that in the system of sialic acid, there are many human-specific changes that have occurred. They're related to all kinds of phenomena that are unique to humans, from disease to running to aging to all kinds of things.
So, I mean, are we better off having that incapacitated CMAH gene, or do you think if we revert the genotype to functional CMAH gene, would we also develop resistance to many of the infections that chimpanzees seem to be resistant to, like malaria?
Yeah, it goes both ways. You know, there's no good and bad in evolution. It's what happened basically. So, and the other thing about evolution, I point out to students, is you can't go back and get it back. I can't say it. So evolution is like a motor car going full speed down the highway, and you have to fix things. You can't stop. You can't pull over and say, I'm going to stop for a little bit and adjust myself. No, you got to keep going. You'll change while you're going. Meanwhile, suppose you had to throw out your windshield wiper for some reason. You can't go back and say, can I get that again? It's gone. So asking that question is sort of like, there's no experiment to answer that. But right now it doesn't occur in humans at all.
Well, do you think that with Jennifer Doudna's CRISPR technology, it's possible to refunctionalize the CMAH gene? And I mean, I guess the experiment would be unethical to do. I mean, we can't do it.
Not only unethical, we end up with lots of problems with immune responses, because it's a foreign sugar, basically.
Right. Yeah, that's true. Yeah. So I would like to now move on to the next subject. I mean, do you want to talk about Siglex? About the glycobiology?
Siglex, basically, what I tell you is that we all know about dangerous molecular patterns and pathogens in molecular patterns. So I asked the question: how do we know self? Why don't my neutrophils attack me? Turns out there's an entire system of proteins that recognize sialic acids on my surface and say, okay, leave them alone. It's part of me. So that is the Siglex are primarily involved in that. They involve many other things. So we discovered and named the family of genes, and they involved in self-recognition. So self-associated molecular pattern. And I do many other aspects of that, yeah.
So these are recognition molecules particular to a specific individual. This is not to a species. This is, you're talking about...
more species specific.
Species specific, yeah. Okay.
But a mouse and a human is quite different in terms of...
Yeah, okay. So now a very interesting topic, your book, Denial, right? Mm-hmm. Now, it's not just your book. You co-authored it with Danny Brewer, who you met by accident. And he doesn't even work in your area. You just happen to be sitting next to him, and he doesn't work in the area in which the book is written about, neither do you. But he had a hypothesis. He shared it with you. You were intrigued. You followed up on it, but he had passed away. And you spoke to his widow and got the manuscript, his unfinished manuscript, and you completed the book. You probably fleshed it out a lot more and made a complete story out of it. Now, can you tell us the core thesis and behind Denial? What is it you're trying to say in Denial?
Right. So just to recap the background, I got interested in what makes us human, which is the question of anthropogeny. And I happened to give this lecture, and as you said, I sat next to Danny Brower, he gave me his idea, and then found out that he died. And so I took his idea and expanded it further with my knowledge, because by the time I was really heavily into saying what makes us human, put it together. So the basic idea is this. Danny said, you're asking the wrong question. You're all asking what made us human. You should ask what stopped everybody else. You got elephants, you got crows, you got dolphins, you got all kinds of amazing creatures around, extremely smart. Why haven't they taken over the world? Why are we destroying all of them and they're so successful? He said, you should be asking what prevented us from getting there. Now, why don't you have 10 different human-like species? The situation you have now with humans, something like the bears all over the world, cross-fertile. The situation in humans is like as if the grizzly bear in North America took over and replaced all the other bears. And so it must be something special to get past it. So Danny said you should be asking what stopped everybody, not what took us forward. The more I read about it, I realized that he was talking about something called theory of mind, which right now, I'm talking to you. You can put yourself in my brain. I'm putting myself in the brain of 92 people that are out there listening to me. And so we can communicate from my mind, or I can say Sherlock Holmes, and you still know what I mean. I'm talking to somebody that doesn't exist. So this is called theory of mind, and it should be a good thing, right? If you have theory of mind, you can tell lies, you can take over, you can dominate. But think about it when it happened. The first time it happened in evolution. It happened many times, but never goes forward. Let's say you're an elephant that's got this ability to read the minds of the other elephants. Great, right? You can take over, you can dominate. That's not the case, because you see a friend get killed in battle. You say, oh, that person is a person just like me. I'm going to die. And so you're going to run away. Or let's say a woman who watches her sister die in childbirth. Oh, I don't want that to happen. I know what happened to her. So you wouldn't go anywhere. So the idea is that the first time the theory of mind emerges in a way that's really functional, it's actually bad news, because you have fear of death. Most of the animals barely understand death, and most don't understand it at all. So once you got there, what are you going to do? You're going to stop there. So the way around it is simultaneously develop some ability to deny reality. So ignore it. I'm going to die, yeah, but actually right now, look at all of us. We're sitting on, we're all headed to a precipice of climate change, dramatic climate change. It's going to be, no question, climate catastrophe is coming. We're all sitting there saying, oh, it's okay. Chalta hai, it'll be okay. Somehow we'll get away with it. And we were all in denial, basically, about the reality. But that denial allows us then to cross this barrier of not being afraid of death. On the other hand, what is optimism? Optimism is denial of death. Optimism is denial. What's extreme optimism? Extreme denial. When I was growing up, my grandfather was a friend of Gandhi, so I often heard about this great man. What is this guy thinking? He thought he was going to take over the British Empire with non-violence? Crazy idea. Totally out of reality, but he paid the price. But we then got Satyagraha and all the other great things that labor movements and all those things. So basically, it's a combined, complicated explanation for one. The only right now known explanation that can explain this. But there's a problem. As you said, Danny Brown is an insect geneticist, and I'm a physician scientist. Who the heck are we to talk about this? We don't have expertise in this. So basically, it's been largely ignored by the mainstream of. And if I had the time, I'd go back and bring it out, and I'm still convinced it's totally correct. I've got too many other things to do right now. But the bottom line is, I think we all have this ability to corrupt reality.
Yeah. So that's it's an interesting. It's still a hypothesis, I guess. It's
right.
Not. And it's possible to prove it.
It's convinced. It fits all known facts, but you cannot prove it.
Yeah. So one way. I mean, would you say that the Neanderthals did not have this fitness? They were not in this fitness landscape, and we were in the fitness landscape. We displaced them. Would you say that could be a hypothesis?
That's what I think. But it's very unpopular to say that Neanderthals were inferior to us. There's this thing called, anything. I think the anthropocentric people they call you out for it. But humans are unusual. So Neanderthals probably did not have the full theory of mind. They're probably just sitting on the edge, crossing over, but they never crossed over. Yeah, there's a few exceptions. Neanderthals never buried their dead. And even they
did not bury their dead, right?
Even when they did, in a few cases, you don't have flowers and artifacts. But as human burials, by the time of 100,000 years ago, you have a woman buried with a dog later. And so you can imagine that this species thinking, oh, that was a person like me, not the other species.
I mean, we can go on and on about talking about this book of yours and this hypothesis.
But just to be clear, the book is not very well written, because they took somebody else's book and tried to finish it fast. So there's actually one or two articles. If you go to my website, you can find more recent write-up about it. But basic theory holds.
Okay. So, I mean, connecting this topic to something we were talking about before about physicians becoming scientists, I think these kinds of polymathic big ideas, physicians are very comfortable with that. Yeah. Because having conquered the entire human body as a medical student, I mean, you dissect it, you understand the physiology of the whole body, you use tools like drugs to manipulate the function of the body, and then you acquire a certain feeling of, a grandiose feeling that, yes, I'm capable of anything. and you lose the fear of entering into new areas. I think this is an advantage and a disadvantage, perhaps, right? Some physicians may stray too far out of their area of expertise and end up making fools of themselves. But it is necessary for people like that to kind of go out and say something that nobody else is saying, right? We also need innovation in thinking. So I guess this is where a physician doing science helps, because they are able to break those mental barriers that a non-physician may have.
Yeah, I think you hit the nail on the head. Going to medical school is like drinking from a fire hose.
Yeah.
It's impossible, so much. But once you've been to medical school, okay, I can go learn something. And I came across this glycobiology, sialic acid. I had no knowledge of it at all.
Yeah,
it's okay. I did so many other things. I can do that.
Yeah.
So it's what the Nobel laureates Brown and Goldstein, who are also MDs, medical doctors, say. They call it technical courage.
Mm.
You're not scared of going into another field. So I've been able to wander between fields.
Yeah, and usually it is the space between fields where the most interesting things can be found.
Exactly.
Right. So that's.
But you have the problem that at the time that this first discovered, most people ignore it.
Yeah.
Whether it be, like you can do a whole long list of things: Semmelweis and antisepsis, Alfred Wegener and the plate tectonics, and any of those things. You find that they were ignored in their lifetimes. Copernicus and.
No, but you published in Nature and things like that. Have you published
I published a little article about what they were trying to encourage me to do. The publicists, they wanted me to go on TV shows and go and hawk the book and talk about it like people normally do. But I wasn't prepared for that, because that's wasn't my intention to even do it, and I just happened to run into it. But I'm not sure. I might take the time, to go back to it. But then it ended up with so the other thing happened, because I needed information. I started talking to people from all over the world. That ended up in the Center of Academic Training. In anthropogeny. And so I run the center with 400 people across the world, all interested in this topic: linguists, biochemists, mathematicians, everybody.
I mean, it's still a very interesting question as to how.
So I forget this Saturday is the next symposium. If you want to look it up, it's streaming live. Please
share with us a link or whatever. I would personally like to attend, and I'm sure.
It's actually all those symposia we organize get recorded and put on YouTube, and now we've just hit 40 million hits.
Okay.
Symposia on all topics related to human origins, basically.
Yeah. So, final question: are you working on the applied aspects of your science, in the sense that, is there a drug, a treatment, a biotech company in your future? Are you working with pharmaceutical companies to see whether anything can be productized?
Before I go any further, if I was remiss in not pointing out that my wife, Nissi Varki, pathologist, excellent scientist, if she was not there behind me and working with me on these projects, I wouldn't be anywhere near where I'm right now. And sorry, go back. What were you saying? You're asking about?
I was asking about.
So Nissi and I started a company about sialic acid biology and cancer, and it's now got antibodies in trials, which we're no longer involved in the clinical aspect. We get out of it once it goes there. And there's another thing we're working on, which is the fact that this non-human molecule can cause immune reactions in humans. When you eat red meat and so on. So we're working on that. And there's several other projects going that have applications. But recently I just wrote an article about sepsis. Not my field, but because I'd seen patients with sepsis, I could see the importance of pH and zinc and so on.
So that's, I had a question about your collaboration with Nissi Varki, because obviously that's worked very well for both of you. And I heard you speak in an interview that when you come back home together, you don't talk science, but the next morning when you drive to work, you talk a little bit of science. So I think you have worked out that relationship very well and very productive for both of you. So I do have a few questions from people who have asked them before. So one question was, I think you kind of answered that, intracellular protein glycosylation and how it affects cellular physiology. Do you have anything to add to what you already said?
Yeah, so intracellular, on the cell surface, you have big complicated branches of sugars. Inside the cell, you have one or two sugars, one sugar N-acetylglucosamine. Jerry Hart discovered that. And it affects all kinds of activities throughout the cytoplasm and nucleus. It's a single sugar. They don't have big branch chains. It's like phosphorylation in a way. and it's highly conserved. If you knock that gene out, you basically, it's embryonic lethal at the very earliest stage. So that's highly conserved. Think of this as inside the cell is highly conserved, needs to be, right? In other words, you won't have a cell. Outside the cell, you get complexity and more complexity. So it's definitely intracellular glycosylation.
That question was from Neelab Datta. If you don't mind, I'll mention the names of the people who asked the questions. Neelab Datta from Ashutosh College, Calcutta University. Another question from Shilpa Swaminathan, no relative of mine, from Brisbane, Australia. You have worked with many nationalities, and you continue to work with people in India as well, right? So she asked the question, what are the cultural peculiarities of different nationalities and how they practice science?
Well, that's a difficult question. It's individual. So, each individual is so different, and so I think that changes with time. You know, right now the philosophy of science in the US is very different from what it was back when I started. And the philosophy I'm in favor of is one that says, go and find how things work, and everything else will fall out from there. And try to take the advanced edge of what we don't know about how things work. But if you want to do the same thing everyone else is doing, then you're not going to gain a whole lot.
So you are in favor of curiosity-driven science rather than
very much.
That is surprising coming from a medical scientist, right?
Yeah, actually,
we think of disease and how can cure disease, but you're saying don't worry about that. Just go find out how things work.
No, and then it'll come together. Yeah,
it'll come together.
I think Isaac Asimov said, great moments in science are not Geronimo or Eureka. That's funny.
Yeah.
Usually what happens, you say, that's funny, you don't follow it. It turns out that's the most important thing. You know?
I didn't expect that. Yeah, correct. So how do you, I mean, you have a remarkable career. I mean, I don't know the details I've seen your CV, but you must have had some failures, right? You must have had disappointments. How have you dealt with it? I mean, especially in research, you can have a failure because your hypothesis doesn't work or your grant application doesn't get funded or something of that nature. So how do you deal with that?
You just have to persevere, basically. I mean, there's no other way around it. If you believe in it, you can follow it through and you'll come in the end. But yeah, it's difficult to you put up with some. I mean, there's actually one time, a simple thing, I was working at the Confell lab, and one assay that I was doing stopped working. Just stopped working. 6 weeks it didn't work. I even went to the next building and got water from the next building. And I went back and looked and realized there's one small change I had made. But I was so frustrated for that time. But then after that, I was able to put together a paper in a month. So we scientists, we live for that moment, you know, when something suddenly breaks through.
Yeah. And so I don't have any more questions from what was asked. So let me go to the questions that are being asked live, right? So one of the questions is, what, in your opinion, would make a medical student want to take up a career in research?
So to be curious about how things work. In other words, in medicine, we have a lot of ways of doing things. Many times we don't know how it works, why it works. And I think it's that curiosity that drives you towards science. But it's a hard balance. I mean, it's become more and more difficult for somebody to be a physician and a scientist. But I think the training as a physician is going to help you do good science, that's for sure.
So, I mean, I have also been through the medical curriculum in India, and you have been through the medical. So, and you mentioned this earlier. You said medicine is an art; it is not practiced like a science, right? So that is one of the frustrating things for somebody who is science-oriented, evidence-based, based on empirical evidence rather than somebody's gut feelings, right? So, in a sense, if you spend 6.5 years training in medicine, it draws away all your curiosity juices. It makes
because you have to learn something for the test next week.
And so only at the end of the 6.5 years, maybe 1% or 2% of the class still have the energy and drive to go and want to do new things, explore, ask questions and answer them in an objective, experimental fashion, as opposed to kind of go by subjective feelings. So given that, what do you think should change in the undergraduate medical curriculum so that more of us retain that energy and interest in curiosity-driven interest in testing ideas?
I think he should. I mean, it's always going to be a minority, no question about that. And it's a good minority of people, but you don't need that many. You know, good people who have crossover. So I think those people are just going to have to focus on the things they're interested in, and go through medicine, and then eventually find out if they can work in science. But it's not a given. But another important point is that even if somebody goes through this and quote fails, and they go back in the clinic, it's still actually a wonderful thing, because now you've got a brain in the clinic that knows a lot of science.
Yeah,
and that can pay off, you know. So
no, one of the bugbears that I have about medical education still in this country is the way we have this 3 years, 2.5 years of preclinical, and then you go to the clinical, and then you forget all your preclinical, and you're kind of suddenly doing patient care, and you've forgotten your pharmacology, but the physiology and anatomy, right? So how is it in the US? I mean, do you have a more integrated curriculum where you are taught all these basic sciences all the way into your late clinical years, or is it still segmented in those kind of fashion?
It's sort of a hybrid. Depends on the institution. Some institutions have very strong, you know, overlap. Others don't. But you know, it falls both ways. If you want to learn about T and B cells, you learn about it in the context of a disease, you're more likely to remember it. On the other hand, if you need the time to focus on the science, you may not be able to have the time to also integrate at the same time. So I think of it like a jigsaw puzzle. There are pieces you're filling in. At any one time, you don't know which piece is important. And then 5 years later, realize, oh, because I saw this one patient who had this one problem, and I saw this other result in this other paper, I can now think of a new idea. And so I think you can't really plan it very accurately that way.
Yeah. So get a little bit extracurricular with respect to your life, right? If you had not been a physician scientist, what would you have been? Would you be a musician or a painter? What interests you?
Apparently, according to my mother, I told her when I was 4 years old I wanted to be a doctor.
Okay, so it was.
I guess I already decided. So actually, the funny thing, my grandfather said, "So why are you doing mathematics?" So I can pay my bills, get my bills. So I always wanted to be a physician. It just the other things came along with that, right?
So now, do you miss being a physician now? In this
I miss the clinical aspect of it very much, but those have become so complicated now and bureaucratic in a way. Electronic medical records and all those things. So there isn't that much contact with the patient anymore. And although we've done great things in science, the medical aspect is, you know, still waning. So I'm not sure I'd want to see patients actively now. I wouldn't mind meeting patients, talking to them, listen on case reports and things like that.
So there were a couple more questions. I'll try to get to them. So there are a lot of questions about your entry into glycobiology. I mean, clearly your entry was smoothened in a way because you're working with Confel, right? So I'm just imagine a scenario where you didn't work with Confel, you worked with somebody else. Would you have been doing something completely different then?
Completely different, yeah.
Yeah.
That's the other thing about you cannot predict, you know, what you're going to end up doing. You may end up discovering something that had nothing to do with what you started out from.
Yeah. So science is in a sense. Science, not science, in your life in general, I think it's a series of accidents, right? I mean, you series of coincidences, accidents, serendipity. You meet somebody, for example, you're meeting with Danny Brewer. I mean, you didn't plan that. It just happened. You happened to be sitting next to him, but from that, you have created a body of work in a book and several articles and an organization, Carta, right? So you didn't plan that. It just happened because you happened to sit next to somebody who had a very interesting idea. So in that sense, science is more like, it's not like Lego bricks. You can't just assemble a career. You just build a series of competences and you expose yourself to many different experiences, and some of them will carry you all the way to where you want to be. And many of them may not. I mean, there will be disappointments along the way, but you need to, I would imagine somebody like you, I mean, you didn't have to, in a sense, you had a sweet career. You didn't move around a lot. You've been at UCSD for how many years?
40.
Yeah, that's incredible. I mean, that's incredible by any standard. So, but nowadays, I don't think it's possible to do that. I mean, you getting tenure is tough. So you apply for grants and then you go where the institution may give you some startup seed funding. So, I mean, would you argue in favor of a more dynamic career where you kind of try many different things in a short span of time?
Yeah, if that's possible, that'd be good. By the way, going back to the analogy of the wall and the bricks, you're right. If you get a brick that sits on top of the next brick, you found something great, you get a good paper.
Yeah.
Let's say you find a brick that's up here. You have no idea what the heck it is. Not connected to any other bricks, right? That's the other thing in science. But somebody needs to pay attention to that brick that's just found there, not just build up, from the bottom up, which is what you do for practical reasons.
There is a question about the fact that, this is something this person has read about you, I think. You have investigated a range of diseases like sepsis, Alzheimer's disease. You worked on Alzheimer's?
Yeah, sure.
I was not aware of that. So infectious disease, cancer, cardiovascular disease, etc. How do you have the courage to explore so many diverse fields in which you may not have a lot of experience? So that's an interesting question. I mean, we already kind of addressed that. Being a physician scientist gives you courage, right? But you must be facing a lot of criticism from peers who say this guy is kind of moving from one to the other. He's trying to do things where he's not been there for more than 6 months and he's now publishing a paper in that area. How do you overcome that?
Well, so my response is I actually work on a single oxygen atom, difference between two sialic acids. So actually I'm very focused in terms of the chemistry. I focus only on sialic acids. So, but then I take that in a different field. And we've got a lot of new findings about aging and all kinds of other situations, Alzheimer's disease. And, people often ask, what are you doing in this field? So I have the advantage that I can discover new things.
So one year you may attend the Society for Neuroscience meeting, another year you're attending the cardiology conferences. So that makes for an interesting life. So what is your, now what do you think about science in India? I mean, you visit here, you have connections here. Are we doing 70% right? I mean, 100% right is impossible, probably you in the US. What percentage of what the Indian establishment in science is doing is right, they should continue to do? And what are the key things they should be doing differently?
Unfortunately, I don't know the system really well, even though I visit regularly and visit institutions that spend time with students and faculty and do science or medicine. I really haven't gotten into the academic, funding system. I don't really understand very well. So I can't really comment in a good way about one way or the other when things are headed.
But can you say what is going on? What is it in the US or any country where you think science is being done very well? What is being done well? Where is not important? What are the things you feel are excellent practices for a scientific establishment to put in place?
So one thing I noticed is that I get applications of postdocs from all over the place. Right? If they're coming from Argentina government supports them to come to the US to get trained and go back. They come from India, is much harder.
Okay.
So I think that's one thing that some countries do, that they make sure that people go out and get trained, but there's a plan behind that. In India, at least for the time I came, it was just random, random interest. So I don't understand the whole system, so I don't want to say something that might be wrong about the Indian system.
No, I was just asking as a general question. So I know that there are lots of scientists in the US who are very frustrated with the funding situation there, etc. So, and there is always a clamor for more funding, right? In every country, scientists want more funding for science, and which is probably needed. But there is also the issue of quality of science being done, right? So you can just put lots of money into science, but I think people should also think about what is this delivering for society, because this is taxpayers' money at the end of the day.
Right.
Is that a question in the US? I mean, are they talking about?
Yeah, it's become more and more of a question, and it cuts both ways. On the one hand, it's right. It's taxpayers' money, you should be doing something useful with it. On the other hand, we're doing basic research. People come and say, what's that got to do with anything? Why are you studying worms? It's not going to worm. And yet that has a lot to do with human biology in the end. So it's a mixed situation there.
And lots of papers get retracted. You have a lot of papers getting rejected, which means some of it could have been avoided by just doing quality science.
That's a problem. The entire peer, having been an editor for 5 years, JCI, I know the peer review system has really deteriorated badly. And there's all these different stupid impact factor, all these different ways of assessing things. And really, I think the system is broken down a lot. And now there's all these junk journals showing up everywhere. Everybody wants to write a paper, so you can find papers appearing that maybe totally made up or just copying something else or everything else. Yeah. The amount of misbehavior in science has increased a lot.
Yeah.
That's because of the pressure to publish and do things. But I think that's just a problem I see.
Yeah. So, I don't want to leave us on a negative note.
Sure.
But there are lots of good things happening and things are improving. One of the things that is becoming better, especially after this pandemic, is international collaborations. I think there is a lot more visibility for science in India outside. And I see the young group of scientists in India, some of them are really, really good and they deserve to go places. And we at Ignite, that's our mission. We want to make science in India go places in a selective fashion, not across the board because we cannot afford that. So I will end here thanking you for having taken the time at a 6 a.m. time. If you want to say something in conclusion.
One last thing for young people out there. So like you said, it sounds a bit negative, things I said, but think about it this way. When I get up every morning, I can't wait to go to work and find out what's happened in science. Most of my classmates are trying to retire from medicine. So the one thing about science is you get up every morning, you want something new. So it's very satisfying.
Yeah.
A lot of frustration, but it's extremely satisfying. Most of the occupations are not. Being an orchestra conductor is something like that. But.
And you also look very young. So obviously that positivity has helped you stay young. So thank you very much, Ajit.
Thank you.
It's wonderful. I hope we can do this sometime in the future as well, because there's lots more to cover. I'm sure you're going to be doing a lot of excellent science we want to hear about. I thank everybody who attended. I'm sorry that we can't take every question, but I'm glad that many of you did ask questions. We could get some interesting discussion going with those questions. I promised to make a couple of announcements. I think one announcement first of all, this is a bi-monthly event. Bi-monthly meaning that we do it once every 2 months. So the next event is in April, and we have Ron Vale, who is head of Howard Hughes Medical Institute in the US. And he's also very familiar with the Indian scenario. Many of you may have heard of him or know him. So he will be our next conversationalist, and we'll find out who's doing it from Ignite. It could be me or somebody else. And the other thing is we have announced that we will be giving out 2 prizes for the 2 best questions. We will do that in a few days. Tomorrow is Mahashivaratri. None of you are going to sleep tonight. But so neither will I. So, but we will in 2 or 3 days time, we will make an announcement. Please visit ignitelesf.in. That is the website of the company I work at, the company that is co-hosting this event along with Cactus and IndiaBioscience. Our website will carry the announcement about the prizes. So if you want to find out if you or somebody you know has won the prize, please check our website and find out. So thank you, everybody. It's been great doing this. Thank you very much again, Ajit. I hope we have the pleasure of meeting. If I by accident sit next to you on a flight, you can be sure you will not rest. Okay. Bye, everybody. Thank you all. Bye.
Thank you.